So a very small portion of the genome is responsible for our identity or uniqueness. That's why if instead of understanding or reading the whole DNA, if we only give attention to that variable part, it helps us to identify the person. That's why it is called as DNA fingerprinting or DNA profiling. So the 0.1% or 3 into 10 raised to the power 6 base pairs out of 3 into 10 raised to the power 9 base pair provide each one of us uniqueness. So we are different from each other because of something which is different in this much part of our genome. So compared to the DNA sequencing, DNA fingerprinting is a very quick way because you are not going to read the whole DNA, you are going to restrict yourself to a particular portion, and that is just 0.1%. And even out of this, we don't read the whole 0.1%, a particular part of this only. So compared to this DNA sequencing, this process is a faster process to find the genetic difference between two individuals or among individuals of the same population. It involves identifying differences in some specific regions in the DNA sequence, and those sequences are called as repetitive DNA. In these sequences, a small stretch of DNA is repeated many times, and they can be separated as satellite DNA from the bulk genomic DNA during the density gradient centrifugation. If you read this statement from the NCERT, it also talks about the similar thing, that when you centrifuge the human genome or the DNA of human, you will find that there are different peaks developing because of centrifugation. Those peaks are because of different density. So density gradient centrifugation means that cesium chloride again will be used here, and when you do the centrifugation, there will be separate satellites developing which is separate from the bulk genomic DNA. So the major portion of the genome will form a single bulk because of a particular density is there, but then there are smaller peaks or smaller groups of the genome will also develop there, and those are called as the satellite DNAs. Now satellite DNA will be of different types. So the NCERT statements I have mentioned here, depending on the base composition. So a satellite DNA can be AT rich. We all know if A is present on one strand, then the opposite strand will carry T, and similarly G and C. So a repetitive DNA can be, or satellite DNA can be rich in either AT or it can be rich in GC, and then the length of the segment, that means it can be 5 base pair, it can be 6 base pair, it can be 20 base pairs long. So whether it is rich in AT or GC, or whether how long is the length of this repetitive DNA, and then the number of repeats also. Accordingly, the satellite DNA can be classified as microsatellite or can be classified as mini satellite. So if you remember, I have already covered this in the last class also. So this is the topic, that it can be mini satellite or it can be microsatellite. If it is 11 to 60 base pair long repetition and the repeat numbers are up to thousands, then it is called as mini satellites. And if it is a repeat which is made up of just up to 10 base pairs and then it is repeated 10 to 100 times, it is called as microsatellites. And I already told you in the last class, 30,000 places have been discovered in human genome where mini satellites are there, and 200,000 places have been discovered in the human genome where microsatellites are there. So let's read the next point now. So depending upon the base composition, length of the segment, so it can be the length can be 11 to 60 or length can be up to 10. And then the number of repeats, the repeats can be 1000 time repeats or it can be 10 to 100 times repeats. They are called as mini satellites or microsatellites. They do not code for any protein. So remember, they do not have any direct coding, but they form a large portion of the human genome. Satellite DNA show high degree of polymorphism. So we'll be learning today now what is polymorphism, and form basis of DNA fingerprinting. Now let's first of all read from here, then I will explain. Variations we all know arise because of mutation. So you need to remember this. Satellite DNA and the polymorphism found in this satellite DNA, this is the basis of DNA fingerprinting. So how do we perform DNA fingerprinting? Because each one of us will have polymorphism available in our satellite DNA. This is the basis of DNA fingerprinting. So now what is polymorphism? Let's understand this. So there are variations we know in the which occurs because of mutation, and now therefore DNA polymorphism is the occurrence of mutation in a population at a higher frequency. So let me now go through the NCERT, because NCERT has given a very simple method of explaining this concept. Page number 121. Let me read from here. So the NCERT also talks about same thing, that the polymorphism is the basis of the DNA fingerprinting. So as polymorphism in this DNA sequence is the basis of the genetic mapping of human genome as well as of the DNA fingerprinting, it is essential that we understand what is DNA polymorphism in simple terms. So what is poly? Poly means more than one, that means variation, and morphism means the physical things. So variation which are available at the genetic level are called as the polymorphism, and these variations occur because of mutation. Now we have already covered this topic in the mutation in the chapter number 5. So new mutation arise in an individual either in the somatic cell. Now we all have done mutation topic before also. Mutation can arise in the somatic cell or it may arise in the germ cell. If it arises in the somatic cell, what will happen? With the death of the individual, mutation is gone because it does not pass on to the next generation. But if a mutation arises in the germ cell, so what are germ cells? Cells that generate gametes in sexually reproducing organisms. So if a mutation arises in the germ cell, then what will happen? It has the tendency to pass on to the next generation.